Thank you very much, and I too would like to thank the organisers for inviting me to speak this afternoon. I'm talking about some of the work that we've been doing over the last few years, looking at mechanisms relating to POLG disease, polymerase gamma disease, and how we are trying to model these in stem cells. My disclosures: I'm now a member of the Oversight Committee for Renio Pharma. I was on the Adjudication Committee for Stealth. I work as a professor in the University of Bergen and a consultant neurologist, and I also work for a specialist centre in the University of Oslo in mitochondrial medicine. What I'm going to speak about this afternoon is really to show you the clinical work and how we're trying to extend the work from the clinic into stem cell models. The work is entirely focused on polymerase gamma disease. It's in the disease state that I'm going to talk mostly about is the disease affecting the brain. And we will review some of the studies that we've done in using our POLG registry of patients and our clinical studies and post-mortem studies to set the scene for how we're trying to model POLG disease in stem cells. This is the clinical paradigm of POLG gamma, and as most of you know, there's quite a wide clinical spectrum, and it can affect different tissues at different ages. And if you can see my cursor here, we see that early onset diseases such as Alpers affects both the brain and the liver. And then there's this tranche of diseases affecting the nervous system, presenting with encephalopathy and epilepsy and ataxia, before the later onset disorders of PEO and ovarian failure. So a complex disease made more complex by the fact that we are very good at describing syndromes, and many names have been added to the POLG phenotype, including one that we gave the epilepsy encephalopathy, which is mitochondrial spinocerebellar ataxia and epilepsy. But there's a raft of others, which makes it clinically quite difficult to follow. We've been spending a lot of time looking at our patients in the registry, trying to simplify this, and that's the first part of what I will talk about. We've been running a national registry now for it's difficult to remember, maybe 6 or 7 years, and we have over 80 patients from Norway, and we've included patients now from Scandinavian countries, which are part of the Mitochondrial Clinical Research Network, which was established more than 15 years ago, but now also includes some of the Benelux countries, the UK and Spain. We've got over 180 patients in this registry now, both living and dead, and we're slowly going through the data, trying to ask questions that are relevant for understanding this disease. When we did this recently, and this is published work, we found that instead of there being neat distinctions between phenotypes and age groups, that most of the features of POLG gamma disease form a continuum, so that if we look at seizures, they can occur at any age, but the median age is young. If we look at peripheral neuropathy and ataxia, then these patients are presenting mostly later on, but can present very early as well. So one of the key features of the publication, looking at these 155 patients, was to say that POLG isn't nicely split up into syndromes, but in fact is a continuum of various clinical features. Having said that, we then looked back and looked to see whether we could group these features, and we see that there are definitely features that occur early, there are definitely features that occur late, and some of the features occur in the middle. So there are age groups in these that we can define, and when we looked at the cluster analysis, it becomes a little bit more complex because the features are distributed all over. But essentially, we can group syndromes around age groups. And I think this is much more useful because it means that we can tell our clinical colleagues that in their age group, these are the primary features that they should be looking for that should make them think of mitochondrial disease caused by POLG gamma mutations. On the same data, we looked and repeated some of the work we've done much earlier, looking at what were the prognostic features and what genotype-phenotype correlations we could make. And it's quite clear from even from this larger material that the key factor in prognosis is the presence of seizures. If a patient manages not to develop seizures, and this now includes all the patients who've got both early onset disease and late onset disease, if they manage to avoid seizures, then they survive for much longer. The other feature that we've repeatedly shown, and now again in this large material, is that the genotype has major implications. Homozygous patients, for whatever mutation, survive better than heterozygous ones. We went further and looked into the epilepsy, and when we looked at the onset of epilepsy, the first seizure, and we looked at all 155 patients, then we see that there's a bimodal distribution. The early onset group who are presenting with what is most probably Alpers disease, and then there's this group here. This is done in months, unfortunately, but this is around 13 to 15 years old. This group here is another peak showing epilepsy. When you when you separate this into genders, you see that this second peak is entirely dominated by females. There are males developing epilepsy all through the age spectrum, but this peak seems to be related to females. So we went back and looked in the data, and females also show worsening around both menstruation and pregnancy. So there is a factor here that exposes females to the onset or worsening of epilepsy, which appears to be hormonally driven. That's the registry. I want to go back in time and look at some of the work that we did to look at how neurons die. And in the brain, at least in our population, the process appears to be an acute and chronic disorder. There's a chronic, slow, progressive neuronal loss or neurodegeneration, call it what you like. And on top of this is an acute episodic phenomenon. These have been called stroke-like episodes, but they're not ischemic, they're encephalopathic and epileptic. So this, and they show up on the MRI as a high signal lesion in the diffusion-weighted pictures and a low intensity, a low intensity band here you see in the ADC map. So this would be ischemic if it wasn't for the fact that this is mitochondrial disease causing this, and this is energy metabolism. And when you get the patient to post-mortem, you see massive changes in these lesions that have occurred with both laminar necrosis and major fallout of the neurons in the area, except for surviving neurons that also show that they're complex 1 deficient. And this is unusual and would be absolutely contra to any ischemic etiology. So I'm quite convinced that these are not ischemic lesions, they're energy-dependent necrotic lesions. And I would suggest that we call them that rather than keep going with stroke-like epithets. So this is what we know about the physical death of neurons. And when we looked in the neurons themselves and started analyzing the mitochondrial DNA content, the respiratory chain complexes, and looked at patients who had died at various ages. So disease duration is essentially how long the patient has lived with the disease. And if we look at mitochondrial DNA depletion, which is done in individually picked neurons, many of them having we had to group them in 5s or 10s, but these are all single neuron picks. We found that mitochondrial DNA depletion was present in neurons, surviving neurons, at the earliest age that we got post-mortem material. This is around 6 or 7 months, and that the level is approximately the same, about 60%. We also see that there are increasing numbers of complex 1 deficient neurons in the brain, surviving brain, and as we pick neurons from these same areas, we see that the number of mitochondrial DNA deletions increases. So this is the molecular pathological signature of POLG gamma in the brain. Having told you a few snippets of clinical and pathological data, the question for us was, how can we go ahead and model this disease, and in which system should we do it? There are a number of systems available, and people choose what they like. I didn't like any of the disease models that were currently available, so opted to go for stem cell models and use induced pluripotent stem cells, which are derived from the patient's own cells. These are then reverted to a stem cell-like state and then become immortal and capable of differentiating into whatever tissue you would desire. And when we started this, we had essentially 3 different genotypes: the homozygous 748, the homozygous 467, and the compound heterozygous, which are the commonest variations of the genotype that we see in Norway. We were able to make homozygous 748 cells and compound heterozygous, but not 467 homozygous cells. So all these experiments are based around these, and the different genotypes are abbreviated to WS5A and CP2A. We took controls at that time from control fibroblasts that were transformed into stem cells, and we used ES cells as internal controls. The next question we had to ask is, what would we do with these stem cells, and what would we differentiate them into? We differentiate through neural stem cells, which are progenitor lines, and we could make dopaminergic neurons or motor neurons or astrocytes or whatever. But the question is, what would be the most robust system to use? The question here is, once you get to neurons, once you've differentiated them that far, they start dividing, and the experiment has to be done on those, and then you have to start again, differentiating them all the way through to neurons before you can perform the next experiment. We chose, therefore, to look at neural stem cells, which are still dividing, can be frozen down and then taken up again to work on. And therefore, we began using these to experiment on and see whether they also manifested the phenotype. The next question we had to answer was what kind of readouts, and of course, all of these dictated by what we found in the pathological studies, i.e. we wanted to look at mitochondrial DNA, we wanted to look at mitochondrial parameters, including the amounts of the complexes and the membrane potential, and we did this in different ways. Some of it was done in live cells using flow cytometry, and some of it wasn't. qPCR and fixing cells to look at other processes that are involved in cell death. When we took the neural stem cells from patients with polymerase gamma, we found that they replicated what we saw in post-mortem investigations. We saw that there was a clear complex 1 deficiency, controls, patients. These are the 748 homozygous. These are the compound heterozygous. And we saw that there was a change in the redox ratio, which we presume follows the complex 1 deficiency. We were also able to show that these cells expressed mitochondrial DNA depletion. There was a lower copy number than the controls. but we didn't find any evidence of mitochondrial DNA deletions, which isn't particularly surprising, given that deletional increase is an age-related phenomenon, and so we weren't really expecting to see deletions. So when we looked at our questions and said what can we model, we can model what I show here, complex 1 deficiency and mitochondrial DNA depletion, but not deletions. We took this a step further and looked and generated dopaminergic neurons and confirmed that they expressed the right markers. They were Th positive, they expressed the dopamine transporter, they had a membrane potential, they had an electrical potential which we could map on patch clamp testing. So these were relatively real tyrosine hydroxylase dopaminergic neurons. And when we looked at these, we saw that they too manifested mitochondrial DNA depletion, low complex 1, and a low membrane potential. Because of the problem around numbers and having to regrow these things, we did the experiment on what was a diminishing number of neurons, and we ended up trying to see whether we could improve any of these features with an antioxidant N-acetylcysteine amide, and we treated cells for 24 hours with 250 micromolar of NACA, and we saw a response, but we couldn't generate enough cells to do the same kinds of examinations we saw with the complex 1 and the mitochondrial DNA. But we saw a response to treatment, which was a resolution of the elevated ROS and a improvement in the mitochondrial membrane potential, which we took to be a positive sign and a sign to suggest that we were improving mitochondrial respiratory chain function. We then became interested because there's a growing interest in the involvement of astrocytes in disease, to see whether astrocytes were involved in the POLG process. And looking at the pathology of it, we do see that there's, in the acute lesions, which we call these focal energy dependent necrotic lesions, there is an astrocytosis, and there's also a microglia activation. So that we looked and we started to generate astrocytes from our induced pluripotent cells. And we managed this, we characterized them. I won't go into all the details of characterization, but then we began to look at the phenotype, in the mitochondrial phenotype. And astrocytes showed both complex 1 and complex 4. Complex 4 was low in the neural stem cells, but it didn't reach significance. But whereas here, they both were low. Complex 2 is normal. There's a drop in the redox ratio that we suspect follows complex 1. And interestingly, the reactive oxygen species, the amount of that fell in these cells rather than rose. To see what was happening at the transcriptional level, we performed RNA-seq on patient controls, and dysregulated genes were mapped, and on KEGG analysis in both of the patient cell lines, the things that came up were autophagy, mitophagy. These were the most dysregulated pathways, and both of them were downregulated. We checked each of the genes, or many of the genes, by qPCR afterwards. This doesn't show up very well on the screen, unfortunately, but using a commercially available kit, we looked at mitophagy and the lysosomal activity in these cells. And in the controls, we could see quite clearly both mitophagy, which is an acidification, and lysosomal activity, whereas in both patients, these were much lower than the controls, and this is shown in the graphs on the right. What we then went on was we tried to dissect what was happening, and it appeared that both the lysosomal function and the mitophagy functions were affected, and also the pink-Parkin pathway was lower in the patients than the controls. So there seemed to be a AMPK related effect going on in both lysosomal function and mitophagy that we thought we could perhaps treat. So we gave the cells, we treated the cells with a combination. We treated them first individually with metformin and nicotinamide riboside, but neither of the two compounds made that much effect alone. Whereas when we treated together, we found that we could improve the mitophagy defect and the lysosomal content in both patient cell lines. And this work has just been published in Frontiers for Cell Biology. Lastly, I just want to tell you an interesting fact that we've observed. When we started deriving astrocytes, patient lines were quite easy to grow. In fact, they seem to grow and be more viable than the control lines. And we performed an assay looking to see whether they would migrate into a damaged area, which they did much more readily than control astrocytes. And then we started co-culturing patient astrocytes with control neurons. And we did this in two ways. We did it indirectly, separated in two compartments, joined only by median, or we made spheroids by adding neurones, oligodendrocytes, and astrocytes together. And in both systems, the normal neurons appear to survive poorly when you have astrocytes with POLG mutations, whereas when you grow them with astrocytes from controls, they survive quite nicely. We're now dissecting this, but it suggests that astrocytes are playing a role in the disease process, even in POLG disease. Lastly, we're clearly trying to develop this system so that we can use stem cells to screen for compounds, looking for new treatments. And we're also on the way to making organoids so we could look at more subtle and complex questions in early disease, particularly in the Alpers phenotype. Just as a summary, if I haven't overstayed my welcome, POLG disease is a continuum, although age permits grouping of patients and prognosis does depend on genotype and the presence of epilepsy. In neurones, complex 1 deficiency and mitochondrial DNA depletion are key features, and they appear to play a role in disease pathogenesis. We can model these features in iPS-derived neurostem cells and astrocytes, and astrocytes appear to be involved in this disease process. The people that did the work, Omar runs the registry and Christian does clinical work. Xiao Liang is the leader of the stem cell group, and Cecilia, you and Ben have been working hard to make these models. Thank you very much.